Welcome to the Human 5.0 Project, the podcast where we cut straight to the breakthroughs that matter, the show where science meets self-optimization. I'm your host, Ultimate Biohacker 10X, and here we break down the real research, real data, and real dosing behind modern biohacking, from compounds and peptides to techniques, devices, lifestyle strategies, and performance protocols. Most people jump in and jump out before they get the gold, so we're keeping it tight and delivering the essential insights you need without the fluff. If you've taken the time to hit play, we'll make sure you walk away with valuable knowledge that can transform your approach to longevity, health optimization, and beyond. Forget everything you think you know about anti-aging. It's 2026, and the loudest shift in regen medicine isn't stem cells. It's what they release. Tiny biological packages that can influence inflammation, repair signaling, and, under very specific conditions, nudge tissues towards recovery. But I'm not here to sell you a fantasy. That happens in gym locker rooms and salons. I'm here to explain what exosomes actually are, what they can do in controlled research, where real human evidence exists, and how to spot hype before it spots your wallet. Hey loyalists, biohackers, and longevity rebels. This is Ultimate Biohacker 10X firing up the mic tonight. We're here to stretch your timeline, bulletproof your cells, and make every extra year feel like 20. And God knows we want at least 20. Welcome back to the Human 5.0 Project. Right now, Exosome Therapy 2026, the next frontier in cellular regeneration, and one of the most misunderstood miracles on the internet. This episode is for educational purposes only and is not medical advice. Exosome products are not FDA approved for treating disease, and many offerings marketed online or in clinics may be unapproved drugs or unlicensed biologics. Outcomes, dosing, purity, and long-term risks are not fully established. If you're considering any regenerative intervention, consult a qualified healthcare professional and verify regulatory compliance, safety, testing, and clinical evidence. Let's dive in. Exosomes are a subtype of extracellular vesicles, or EVs, small membrane wrapped particles cells use to communicate. Inside them, proteins, lipids, and genetic material like microRNA. Here's the nuance most people skip. Serious scientists increasingly prefer the term extracellular vesicles because proving something is specifically an exosome is harder than clinics admit. That's straight from the International Society for Extracellular Vesicles and their MISEV guidelines. So when someone says pure exosomes, the correct response is simple. Show me the characterization. These vesicles don't heal anything by themselves. They carry messages, and messaging depends entirely on source cell, manufacturing process, dose, context. Exosomes aren't magic. They're information. Two reasons this exploded. First, cell-free regenerative medicine. People want stem cell-like effects without implanting living cells. Second, scalability. Theoretically, and that's where regulation matters, because the FDA has issued repeated warnings about illegally marketed exosome products and documented serious adverse events. Translation, the science is advancing faster than oversight. Here's where we get honest. Yes, there is credible lab evidence that certain MSC-derived EVs can reduce markers associated with cellular senescence in specific models. For example, reduced SA-beta-gal positivity, reduced DNA damage signaling, reduced SASP factors. That matters because senescent cells poison tissue environments. But here's the truth influencers skip. EV biology is not one-directional good. Senescent cells release EVs too, and those vesicles can spread inflammatory and pro-senescent signaling. So the honest framing is this. Some EVs may dampen senescence in specific contexts. Some EVs may propagate damage. Outcome depends on origin, content, and delivery. Exosomes reverse aging is not science. It's marketing. Do we have real human signal? Yes, but it's early and condition specific. One example, knee osteoarthritis. A triple-blind, randomized controlled trial evaluated placental MSC-derived EVs versus saline. That doesn't mean OA is solved. It means we've crossed from anecdotes into legitimate clinical investigation. Most claims you see online are still ahead of evidence. Now I'm going to give you a blueprint, not for DIY therapy, but for information literacy. First, the FDA has been extremely clear. There are no FDA-approved exosome therapies, and the agency has issued alerts and enforcement actions concerning unapproved exosome products and reports of serious adverse events. So here's your reality filter. If a clinic can't answer these clearly, you're not looking at advanced medicine, you're looking at a product story. What is the source? MSC, placental, cord, autologous? How are donors screened? Infectious disease, medical history, traceability? GMP manufacturing or backroom compounding? Sterility and endotoxin testing? Let's see the documentation. Characterization per MISEV standards? Markers, particle analysis, contaminants? Potency assay. What functional test is used to show biologic activity? Batch consistency. Are lots standardized or hand-wavy? Clinical evidence. Do they cite controlled trials or just testimonials? Follow-up and adverse event reporting. What's the safety monitoring plan? Regulatory posture. Are they operating under an actual clinical trial registration or an approved pathway? No answers equals no science. The real future isn't mass-market exosomes. It's engineered EVs, it's targeted delivery, it's defined indications, and actual regulation. Longevity isn't about chasing the flashiest clinic. It's about tracking evidence as it matures. Now, I want to end with something that ties this whole story together. If exosomes are the messages, then the next question is, what cells are the best responders to repair signals without the tumor risk baggage? And that's where we pivot into a cell type that has been quietly building real credibility, especially in Japan. That's where we pivot to MUSE cells, MUSE, multilineage differentiating stress-enduring cells. These are stress-tolerant, non-tumorigenic, endogenous stem-like cells with real clinical traction, especially in Japan. And here's the punchline. MUSE isn't just theory. There's been a randomized placebo-controlled trial of a MUSed cell-based product, CL2020, reported for subacute ischemic stroke. There's also ongoing experimental exploration of delivery routes, including intranasal approaches in stroke models. So next episode, we'll go all in. MUSE Cells 2026, the regenerative cell that might outrun stem cell hype. Stay signaling, stay evidence-driven, stay regenerative, and I'm out. Thanks for listening to the Human Upgrade Project. If you found value in today's episode, share it with someone who's ready to elevate their biology. Make sure to follow the show for weekly deep dives into peptides, protocols, devices, lifestyle strategies, and the science of high-performance longevity. Until next time, stay curious, stay optimized, and stay 10x.